Welcome to qnawire.
All right, so this question is a series of fill in the blanks, essentially testing your knowledge of nucleic acids. So, what I'm going to do is I'm going to summarize each of the prompts, and then I'm going to briefly explain why each one is the way it is. So, I would recommend following along with your textbook if you have it. So, for A, it's essentially asking what DNA stands for. And DNA stands for deoxyribonucleic acid. It has a deoxyribose sugar, and it in and of itself is a nucleic acid. Part B says the basic repeating monomers, so essentially the building block of DNA and RNA is known as a nucleotide. In part C, the pentose deoxyribose is found in DNA, whereas the R in RNA stands for ribose, because that is the type of sugar found in RNA. In D, the basic linkage between a sugar molecule and a phosphoric acid is referred to as a phosphate diester, with diester being the key word there. Linkage. And for E, the bases on opposite strands of DNA are complementary to one another, meaning one half essentially perfectly fits with the other via hydrogen bonding. And in F, it is asking what pairs with adenine. And so in DNA, thymine always pairs with adenine. Note that I said in DNA. In RNA, the thymine is actually replaced with uracil. And guanine always pairs with cytosine, whether it's DNA or RNA. Letter G, it essentially asks what a segment of DNA that codes for a specific protein is. That is called a gene. Our genes code for specific proteins, which then correspond to our physical traits or our phenotype. In H, during protein synthesis, transfer RNA or tRNA is responsible for actually assembling the amino acids using instructions coded by messenger RNA or mRNA. And then lastly, the codes specified by the codons on mRNA are responsible for assembling the correct primary structure. So each codon is a sequence of 3 bases, and each codon codes for a specific amino acid, although multiple codons can code for the same amino acid. And so these are all of the blanks for this question.